Hi, guys. Thank you, everybody, for being here today on our Reef Chat about deep sea baby animals with Dexter Davis and Lara Maleen Beckmann. As always, thank you so much for your time. Thank you so much for being here. Quick reminder that if you're interested in learning more about the deep sea, the Sustainable Ocean Alliance has their Deep Sea Academy online. And without further ado, Mr. Dexter and Ms. Lara, the floor is yours.
Thank you,
Hello.
Frida.
I'll let you introduce yourself first, Laura.
Yeah, sure, I can go first. Yeah, thank you for being here. And we are very excited to talk about everything babies in the deep sea. So I'm Lara Beckmann, and I'm a PhD researcher at the University of Gothenburg in Sweden. So it's actually evening here. And I'm focusing mostly on the reproduction and early life stages in corals and sponges in the deep sea. And yeah, I will talk a little bit about my research and also about my connection to the art world a little bit.
And hi, my name is Dexter Davis. I'm an artist with the Deep Sea Biology Society, but I'm also a master's student at University of California, Santa Barbara. And I study informal communities, which are animals that live in the sediment, and how they use methane at methane seeps in Antarctica. But I used to do work studying larval dispersal and settlement in hydrothermal vents and methane seeps. So I'm going to talk a little bit about that work and how it inspired my art as well.
Okay. Yeah, so I already introduced myself. But so I will talk a little bit about my research that I've been doing at the University of Gothenburg. And this project is a North Pacific Research Board project. So I work mostly in the North Pacific with researchers in Alaska. So you have a little bit of a idea where these animals live that I work with. And so a little bit about corals and sponges, because I don't know your background. You might not know that there are actually corals and sponges also in the deep sea, in cold waters, because mostly when we think about these animals, we think about tropical ecosystems, right? Like where it's very warm, it's shallow water, and they're very important for the ecosystems, but the same is true for cold water and deep sea corals. And they are so-called ecosystem engineers. That means they provide a three-dimensional structure on the seafloor. And so with this, they create habitat for other animals, for example, for fish. So a lot of larvae fish or baby fish rely on these structures to find shelter or find food, for example. So it's also nursery grounds for other animals. And so these animals are associated with a lot of other organisms usually. And however, they are also very susceptible to disturbance or damage. So they are quite vulnerable because, yeah, when you trawl them, for example, or if they are removed from their substrate, it takes very, very long time for them to grow back, if at all. So a coral colony, for example, that is yeah, mature or has like a large size can be up to, yeah, several hundred years. Some even say like thousands of years. And the same is true for sponges. And so I'm mostly focusing on the reproduction in these animals, in deep sea sponges and corals. And just to give you a bit of an overview, why why do we need this data or this knowledge is mostly because reproduction is very important for any animal on the planet or for any organisms on a planet because it ensures the continuity of a species, but it's also enables adaptation, for example, to a changing environment. And for any deep sea species of coral, we have so little data. So here, for example, you see a plot with like known species in the deep sea, and all the colored in areas here are the ones where we have information for reproduction. And this is any type of information. So this could also be just like a little bit like knowing, okay, they produce eggs of this size, for example. So it's not very comprehensive. And you see there's very, very little data in general. And I did the same for sponges, just so I know what I deal with. And here it's even less. So we have 15 species where we have some kind of reproductive information. And for glass sponges, which is a different type of sponge in the deep sea, we have maybe 2 species, maybe 4. I'm not even sure if 2 of these studies are really valid. So we have very, very little data for reproduction. And that's where I kind of start with and try to produce more information and knowledge here. So let's start thinking about what a coral life typically looks like. And usually it starts with a mature parent or a coral colony that is mature. And this will be sessile, sitting on the seafloor. And so then if it's mature, the colony will produce gametes, so eggs or sperm. And depending on the species, it can be a single sex or both sexes at the same time. And they can be, for example, released in the water column, or they can also be retained in the colony and be fertilized inside. But here, for example, in this example, in this scheme, the gametes are released in the water column, and then the sperm fertilizes the egg. So it's very similar to humans. And then we have the embryo development. And then we have a very interesting stage that I'm also very interested in, and also Dexter will talk a little bit more about it, is the larvae. And so this planular larvae is typical for corals. It's a very simple larvae, I would say, compared to some other invertebrates. And then this larvae will develop a little bit, maybe swim somewhere where it's happy to settle and find a new spot to settle on. And this larvae will grow to a new adult or a new colony. And this will take very, very long time for deep sea animals or deep sea species. And so we have 2 very distinct or 2 different strategies here. One is the sexual reproduction, where we have the recombination of genes or different contributions. And then the asexual reproduction, which is this part where the colony grows. And this is kind of a clonal reproduction. So the colony will grow by cloning itself. So just a little bit what is possible, because this is kind of what I showed you now is like a simplified life cycle. But there are many different ways how this can proceed. And for asexual reproduction, for example, sponges, for example, they can bud. So that means they just like, they just fragment little parts of the body float away and find a new place. Or polyps, for example, or corals can bail out polyps. That means the polyp is like removed from the colony and then can attach somewhere else. And for the sexual reproduction, there are many different things that can happen. So, for example, as I already said, like a colony can be gonochoric. That means they have separate sexes, so individuals have separate sexes. Or there can be hermaphrodites having male and female gametes in the same individual. And then there are different reproductive modes. So, for example, there is external fertilization, where we have the broadcast spawning, which is something that we think about maybe when we think about tropical corals. What is typical in deep sea corals actually is the internal fertilization. At least that's from the data that we have. It's quite common that the individuals brood. So that means there's internal fertilization, and then they develop the larvae inside the mother or the parent. And so typically that means working with live animals, getting the gametes, for example, looking at the gametes under the microscope, trying to get fertilized eggs and trying to get larvae to work with. So that is one way of doing this type of work. But working with deep sea animals is really tricky. So this is something that takes a lot of time, a lot of resources, and is not often possible because these animals are not very happy in the aquarium or in the tanks. And so, for example, something that we really want to do for deep sea corals is to do these fertilization experiments, where we get the larvae and then do some experiments with the larvae. For example, like how long do they develop, or how long does it take for them to develop to a larvae? How are the different stages of the embryogenesis? And yeah, to just get more information. But this is very tricky in the lab. And so what I usually do is a lot of microscopy. So here are just some images that I've taken over the past years. So for example, I do a lot of histology, which means I slice tissue in very thin sections and then color it. And then you get the different cells, for example, in different colors. And that's how you get some information from the tissue. We also do a lot of electron microscopy. This is this grayish picture here, where you can see very detailed, like for example, cell details. Or also now I'm trying some CT scanning, which is very nice. You can actually look inside the tissue without destroying it. So there are many ways to do these kind of things and get some information at least. For example, for the development of eggs or embryos inside the tissue. Just another example. So here's a coral, and so this is a piece of the coral or a branch, and this is a histological section where you see there are actually some larvae here in the corner, the big ones, and they're very tight together. They're all in one chamber, kind of, and there are also some eggs, the smaller circles here. And these are some larvae, how they look in the histology at least. And there are we can even see different stages. So on the left is a early larvae or planular larvae, and then the late larvae, which is already kind of ready to to leave. It's very close to the opening. And yeah, this is how it looks like when it goes out, probably, but we don't know. And for sponges, as I said, there's so little information. So I'm just showing this because we have very few images of these, and they also have different larvae types. And in the deep sea sponges that I've been looking at, they mostly have these parenchyma larvae that are down here. So they also have some different cells, but relatively simple larvae as well. And I really like to think about the connection between the larvae stage and how this connects to the adults or the species in itself. Because once the larvae leaves the colony or the parent, it's on its own, right? So it swims in the water column, and Dexter will talk more about this. And then it needs to find the perfect place, because this is where it will settle and this is where it will grow. And if it's like if it's just not a good position to be in, it won't be able to survive. And it's so important for it to find a good space to be really able to grow to maturity, for example. So this larvae stage is extremely important. But I will let Dexter continue now.
Right. Yeah, Laura touches on a really important part, is that in this larval stage, this is when these animals are the most mobile. For these invertebrates that are sessile, they sit on the seafloor, they don't move after they are in their adult form. And so when they're in this larval stage, they can swim through the water column and colonize new habitats, but also promote gene flow. This is how new individuals come into a population. It can reduce competition with the adults that already live there if some are going to a new area. And I wanted to talk about it kind of in the context of chemosynthetic habitats, which are areas of the seafloor that rely on chemicals as a food source. So that's methane seeps, hydrothermal vents, whale falls. And these are, the deep sea is already a niche habitat, but these are kind of even more niche in that, there's endemic species that survive there that might even have bacterial symbionts. Basically, they have this relationship with a microbe that allows the animals that live there to get food from those chemicals. And so how larvae move around these habitats is really interesting, and was what the research that I was doing was focused on. I used to work as a research technician for Dr. Sean Ariano at Western Washington University. And so we would go to methane seeps in the Gulf of Mexico. And kind of one of the big questions that we were asking was this top part of this graphic here. And that's, how are these larvae connecting sites that are really far apart? There would be methane seeps that were tens to hundreds of kilometers apart, but you would see the same species there. But we know that these are animals that can't really move around that much. So how are they getting from one place to another? Um, so larvae kind of they don't really have the ability to swim against currents, but they can control their vertical positioning in the water column. So the larvae that is represented here is a bivalve veliger, and they have what's called a velum. And it's basically this sail that has small little hairs called cilia all over it that they can swim with. And so that allows them to go up and down in the water column and say, there's a really strong current up in the mid-water column. They could swim up to that and catch a ride. And in the deep sea, larvae actually stay in this form for a lot longer compared to their nearshore counterparts. So for one species, these Bathymodiolin mussels, their planktonic larval duration, how long they are larvae, is over a year. So these animals have the ability to travel very far. But then there's two other questions that kind of stem from this. As they're moving around, how do they know where to land that they know they can survive in? And for these animals that do have these relationships with microbes, how did they acquire those microbes? When do they acquire those microbes that they need in order to survive as adults? So those were kind of the questions that we were trying to answer in our work. And in the East Pacific Rise and the Lau Basin hydrothermal vent fields, we were doing settlement experiments where we would see, basically we would put two sets of plastic plates down on the seafloor and scrape one off that had any potential microbes that could have been established on them, and basically see if there's more that are settling on these plates that have these biofilms that could be releasing some sort of chemical signature that are telling the larvae, hey, like, you want to land here, this is where you're going to be able to grow up, we've got a great community, we'll support you. And then also by looking at larvae that we collected, we could actually see through their microbiome what is inside of them. So we can kind of see, along that time scale, when are they acquiring these different bacteria. If you want to go to the next slide, Laura. And so these are just two examples of how these larvae can move around. They're not the best quality, but these are both bivalve veligers, so baby mussels or clams. And so on the left here, you can see it's crawling around with a little foot. And then on the right, it's swimming around with its velum. So that's that kind of sail full of cilia. So they have different methods to crawl around too. Through the water column, they're swimming, but when they get to an area, maybe some chemical signature told them to land there, they can physically crawl around and basically look for the most suitable spot. And different larvae can do this differently. Some might also use phototaxis, which is basically an attraction to light, or the opposite, a repel from it. So they can see, maybe it wants to live under a rock. And so, oh, it's too bright here, I'm going to go away from the light. So they have different methods to kind of control where they're dispersing also. Next slide. And so in the deep sea, what Lara mentioned is that there's not a lot of data, and it's really resource intensive to study larvae. We can try to spawn individuals in the lab, but it's really hard to kind of mirror those conditions that are found on the seafloor: cold temperatures, high pressures, maybe presence of different chemical cues. And so the work that I did in Sean's lab, we would try to catch larvae after they were already created, once they're swimming around. And we used all sorts of cool instrumentation to collect larvae. So on the left side there, there's a plankton pump that's attached to this lander, and that would sit on the seafloor and basically suck water through a filter for 24 hours and collect all the larvae or whatever's in the water, copepods, other zooplankton on a mesh that we could sort through later. Or we would make use of submersibles. So here's the remote operated vehicle Jason and the human operated vehicle Alvin. And with those, we could physically collect, sediment from the seafloor. We could take specific water sections, or we could even use what's called a slurp canister, which is basically a little vacuum cleaner, and try to suck off larvae that could be attached to adults on the seafloor. But my favorite was Sentry, which is the one in the bottom right corner. That we called it like the little chicklet because it looks like a little chicken. Or maybe a minion with, giant goggles on it. But attached are these two things called plankzookas, and these are giant funnels that collect whatever the instrument is swimming through. And this is an autonomous vehicle, so it could be pre-programmed to go to a certain depth and basically do any pattern that you wanted. And that's how we would collect most of our larvae. If you want to go to the next slide. And so with those samples, what we would do is we would spend hours sorting under the microscope, whether that's at sea or back on shore. And basically, we were trying to look for target species. If we were looking for those mussels that live at those methane seeps, we might be looking for their larvae or another important species on a different habitat. And I started this as an undergrad. I was definitely not accustomed to sorting larvae. I'd taken some marine invertebrate classes, but what's really interesting about larvae is they come in so many different forms. Lara showed a very basic plan of a planula, which is basically a blob covered in cilia. But a lot of these larvae have they have different methods of swimming. Some have velums, some have flagella. Some are covered in cilia without needing, a sail to fly on. And so I kind of started getting into art through studying larvae as I had to, learn how to identify all of these different larval species. I started to have fun with it and kind of create a larval journal where I basically tried to identify what are the key characteristics for identifying the different larval types to, help me with my sorting, but also because it kind of was just fun to me to see all these different shapes that the larvae had. And one of my friends at the time actually told me that I should turn them into stickers because they're like fun little cartoon creatures. Some people say they look like little Pokemon or something. Um, so yeah, I was inspired by all the different shapes of larvae. And you can see here, this is one entry from that journal I created and two of the resulting stickers from it. You want to go to the next slide? And so these are just some pictures of larvae that we collected during our research expeditions. And you can kind of see the diversity of different larval forms here. In the top left, that's a baby crab. And right next to that, to the right, is a baby clam. Then there's a worm. I think the next one is a planula as well. So that's a baby coral, potentially. There's some worms, a little shrimp, and then the bottom two in the middle are actually baby barnacles. And on the right, this matrix of 9, those are all different shells of different marine snails that we found on one cruise. And so throughout my time in that lab, I think we found 31 or so different larval types in just the methane seep cruise projects. And so I turned them into these stickers, if you want to go to the next slide, that kind of highlight all those different forms of larvae. And yeah, that was kind of how I started getting into art more. I've always been an artist behind the scenes, but then I realized that I could use art as a media to also talk about science. People see these animals and they're like, well, tell me more, what are these? These are real things. There's no way. And then you can kind of, start stimulating those conversations about what larvae are, what they mean, why we should study them. You want to go to the next slide? And so that has kind of led into some of the other art that I make, which these are all community posters from different deep sea habitats I visited. And then the one on the far right is for my current master's project here in Santa Barbara, where I am. And these not only, highlight kind of these communities that a lot of people don't get to see because the deep sea is, challenging to get to, but you can also use it as, a way to um, introduce people to different habitats, but also, that we need to protect them, that they're very fragile ecosystems that these animals are coming from. Um, and so I am really excited to like keep up um, this passion and work with other scientists and hopefully use art as a way to translate the research that they're doing. And I think it's awesome that me and Laura are both on this talk because we're both scientists, but we're both also artists. So we can appreciate the value of the crossover of those two. So Laura, I think now if you want to talk about some of your art as well. Um,
Yeah,
yeah.
it's really nice to see all the different larvae types. The planet of larvae are a little bit, yeah, a bit boring, but they have their values. Yeah, as you said, like I also have a little bit of art background. So before I started biology, actually, I was more into the creative world and was even thinking of studying more into the art, yeah, scene. But then I decided to do science instead. But yeah, I always missed this part of having the creative side, and what I think is really nice to have this combination of doing science and also being creative and doing art on the side. But during my PhD, I didn't really have time to sit down and, draw or do illustrations. And I was working, so besides working on the reproduction, I also do some biodiversity analysis, just like basic studies using videos. So looking at the diversity of corals and sponges in the deep sea. And while I was looking at these videos, I thought this would be amazing to just show an artist and then see what happens. So maybe they get inspired. And so I contacted Kirsten, one of the artists in the Unseen Ocean Collective now, and yeah, we started this project, and then Megan and Nili joined, who are artists, or Megan is an artist from Washington State in Spokane, and Nili is in Oregon, and Kirsten is in Alaska. So now we are, yeah, this Unseen Ocean Collective. So we collaborate together. I give the data, give a little bit of science background, and the artist, yeah, just get inspired by it and can kind of freely decide what they want to create. And just to, yeah, show some examples what has been done. So for example, here on the right is a sculpture from Nili. This is a carnivorous sponge, which was seen close to the Aleutian Islands at 2,000 meters depth. And it was like almost a field of these carnivorous sponges. So all these little bulbs here are structures that the sponge uses to capture animals. So crabs or little crustaceans get caught, and then the sponge will digest it. And in the background, actually, you see one of Megan's drawings. She drew the sponge as well, or painted it. And in the middle is also a painting by Megan. I think she's actually here. Hi, Megan. And she really uses these, yeah, expressive or colorful paintings to really show the color that is in the deep sea, which I think a lot of people are not aware of, that there are actually like really red or purple, pink animals down there. And yeah, these are just amazing paintings to show this. And yeah, on the left, there's a little sculpture that I did. So I also try to contribute a little bit with some sculpting, which is fun to do. And then, oh, I don't have one from Kirsten, but she's doing more, yeah, these scientific natural illustrations, and really showing like the diversity and the details. And so I think with our collective, we really want to show the deep sea as it is. So a lot of people have this image of like vast, deep, dark space where there's nothing really, or if there is something, it's scary and alien. Like we have all these weird words that we usually read when it's about the deep ocean. So we just want to reshape the image of the deep sea and also make aware of how important these ecosystems are for humans as well. So a lot of fish rely on the deep sea. The climate is regulated by the deep sea. So just making aware of it and also showing that actually there's like our planet, 50% of our planet is deep sea, and we never think about it. Well, which makes sense because we never get to it. It's like very difficult to visit it, but it's still important to know about it and also be able um, to have an opinion about it, especially now when people are more interested in resource extraction and doing deep sea mining and all that stuff. So I think it's important to know about it and then have an opinion. Um, exactly. So right now we had our first opening of an art exhibit last week, which was really nice. And yeah, we have some more things planned. So for example, we have another exhibit in Spokane in Washington State for 1 month in April. And yeah, there are more things coming. So if you're interested, you can visit our website and see or read more about it.
I think you bring up a really good point on how we can kind of use art to connect people to the deep sea also and make them have an opinion of it. I think that's one powerful tool of using science and art together is that science can be relatively inaccessible to a lot of people. Reading a scientific paper is not the easiest thing, especially if it's like a huge review, for example. Or even understanding a graph can be challenging at first. But like the work that you and I and the whole Unseen Ocean Collective and Deep Sea Biology Society, all these organizations do, is that they showcase, there are these beautiful animals and beautiful habitats that live down there. Um, and they may seem like otherworldly to some people, but it's very much our world. Um, and yeah, it's making it a lot more accessible to people. There's these live streams that are happening from deep sea cruises or gallery showcases, having Reef Chat. I mean, these are all great ways to bring people to the deep sea and show that it is very important to our planet. And we do need to protect it. And like you said, it's a very crucial time to kind of have that stewardship of the ocean. Because yeah, it's under threat from resource extraction, and we have the opportunity to do something about that.
Yeah, exactly. I also think it's really nice to show these kind of intact ecosystems because that's something like a lot of people think, oh, well, like the forests or whatever, like everything is already um, occupied or a little bit destroyed or it's not natural anymore, not pristine. And we actually have like all these ecosystems that are still very intact and they're really important and huge. So it's also, for me, it's also a source of hope a little bit for like nature and in or yeah, just showing what we still have and what we are able to protect still. Yeah.
I have a question,
Yeah,
I guess. Oh, sorry, Dexter.
go ahead.
In that same vein, like, so what is one thing that you wish people would know, right? Like, if I could give you like a worldwide megaphone to like, that you're like, if people just knew this one thing, like, they would be so much more excited about this. Or if they knew about this one thing, like, it's not about, this is kind of dumb, but I'm always like, if you like pugs, you can find a deep sea creature that you love, you know? Like some of them are ugly cute. Like we have that up here. There's plenty of that down there too. So it's one thing that if you could choose one, which I think it's difficult, what
Mm.
would it be?
That's a tough question. I think for me, like the thing I try to tell to people, I come from an ecology background, is kind of the importance that every animal or every being kind of plays a role in an ecosystem. And so I think, that goes for terrestrial systems too. Like, I know some people don't like insects or spiders and things. And, people are like, oh, if every mosquito disappeared, I wouldn't be bothered or whatever. And while I hate mosquitoes as much as the next person, like, it's still important to recognize that um, all these animals are playing a role. And whether that's, climate management or nutrient cycling, creating habitat for other animals. Like they need protecting because they offer services as a whole, not just as an individual either, but just yeah, having the context that everything is important in an ecosystem. Um, I think is yes for mosquitoes. Um, just yeah, like we should protect animals just for being there. They have an intrinsic value. I think a lot of people are always asking why is this important? Why should I fund this? Why should I care about this? And um, yeah, I think that there's an intrinsic value there that you shouldn't have to like have the reason, but know that, everything is playing a role in our planet. And that's why we want to preserve it and understand, if we are manipulating a system, what is actually going to happen to that system? What are we going to lose also?
Yeah, I totally agree with you, Dexter. I think that's a very good point. And I would probably go with the same notion, like just telling people like everything has a meaning, like that's why we have this diversity. And I would probably add to it, like there's so much diversity, like even for a scientist, like sometimes I read a paper and they say like, oh, we found like these like so many new species. I'm like, what? It's just incredible. Like whenever you go down there with a RV, with a robot, with a camera, and there are probably new species. Like every time you go down, you find a new species, and like we scientists just um, can't continue, or we can't describe everything because we don't have the workforce to do it. So it's pretty incredible when you think about it. And that's not even just the case for the ocean, it's the same for terrestrial ecosystems. So I think telling people about this immense diversity that we have is also, yeah, something that I would focus on. But just one thing I think is really hard.
But yeah, I think you bring up a really great point about deep sea science in general, and kind of this is why I fell into it, is kind of that there is still a lot of unknown there. You were showcasing how much we know about reproduction in different groups, and it's such a small little fraction. And I think, you can look at it at a lens where it's quite daunting to see how much, work there is left to do. But for me, it's exciting because there's so many potential questions to ask. I remember on like the first cruise I went on, I was asking Sean, why is this animal living on this animal? And she was like, I don't know. Nobody knows. Like, that is a potential research question. And I think that makes this field really exciting in that, there's so much potential to still understanding it. I mean, it's still a relatively new field. And I think, as technology has improved, it's increased our access to it. But, like Lara said, there's still, you know, there's a workforce that's needed. So we need scientists also that, want to go out and study these things. Um, and with larvae specifically, since that we're talking about today, like there's a lot of unknown there, Even as we were collecting larvae on our cruises, there would be tons of individuals we did not know what species they belonged to. And even when we did DNA extractions on them, we would not get reads, we wouldn't get identities for them. And so we had to do a lot of like internal matching, where basically we would take adults from these areas and then see if their DNA matched up with the larvae that we were also extracting. Um, so it's really fun because there's so much unknown, and yeah, we're constantly discovering new things. And that's where I think art also plays a really awesome role, is that you can kind of change how these discoveries are translated, by focusing on different aspects of them. You know, if it's a new habitat that you discover and you show these beautiful paintings of corals and sponges that have animals living on them or inside of them, like it creates an even more interesting story that I think people can really like get engrossed in without having to dive super deep into the science behind it, but still have the opportunity to ask those questions, if they feel the need to.
Roxanne, did you want to ask a question?
Yeah, thanks. Hi from New Zealand. I was wondering, Woods Hole did a great immersive art exhibit with Art Tech House, like about the deep sea. I don't know if anyone saw that, but it was super inspiring. And as someone who, like, I love being in the ocean, but as you guys talked about, it's really hard to get to the deep sea. So are there any other art exhibits that have really inspired you that are either like immersive or things that are coming up, or other artists that you haven't mentioned that you think would be cool to flag as folks to watch?
Hmm. Yeah, I think, we kind of talked about these different um, ocean groups. Like there's the Unseen Ocean Collective here, and I'm a member of the Deep Sea Biology Society. And I think a really interesting one to look at is the Deep Sea Biology Society. They have a whole page on artists, and it's very diverse. There's sculptural artists, there's painters, there's digital art, and I know the Deep Sea Biology Society is really creating an effort right now to have more art be involved in the science. So Laura, I don't know, do you want to talk about the artsy matchmaking that's happening right now? Because that's something that's going to come up that's,
Yeah.
I think, going to be awesome.
So I've teamed up with another artist, and we really thought about how we could improve this connection between artists and scientists, because a lot of scientists want to do outreach, but they don't really know how. And a lot of artists want to work with scientists, but there wasn't really a platform to connect. So we had this artsy matchmaking project or yeah, it started in summer last year. And we had this open call, so people could just like fill out a form, and then we connected artists with scientists who fit with like their interests or their topic. And now they're actually in their like active phase, so they started their teamwork or their collaborations. And so in summer we will have a little, well, it will probably be an online exhibit and an auction. And we hope to have sometime maybe an exhibit in person, depending on, yeah, if we can get some funding for it. But that's like a plan to do. And then the Deep Sea Biology Society will also have an exhibit in 2028 in Norway, where the symposium will be. So I think the idea is to have a bigger exhibit there. So there are some things coming up, definitely. Mm.
Yeah,
Cool, thank you.
we're also working with so we have a couple of art residencies where, Lara, if there's another open call, I know a lot of artists that I talked to, they were so sad that they missed it because they were like, how can I do it? So I'll make sure to, keep an eye out because there's a lot of artists that were very sad that they missed the deadline. But we're working right now with a South Korean opera music ensemble, and they want to bring um, a symposium that mixes art and science. And one of the things that we also want to include is a physical art exhibition. Obviously, funding is, we're struggling with funding, but that's the beauty of it, that everywhere around the world, everybody is trying to get the word out there. And so one of the things that I really like to do is not only collaborate, but also lift up art friends and friends around that we're trying to, spread the word, because we're all like little ships in the ocean, and if we all share like charting maps, we're all trying to get to one little island, and there's space for everybody. And so the more people get to know about all the stuff that we're doing and all you guys are doing, the more we might inspire other people to care. And so if anybody else knows of any other exhibitions or artists or anything else, let us know, and we'll make sure to spread the word out as well.
Yeah, that's awesome. Definitely. I think like we had so many artists sign up, so we couldn't even match all of them. So it's really so the scientists were really the the deciding factor. So I think it's also important to encourage scientists to work more interdisciplinary and share their work. So it's still a little bit this yeah, this philosophy maybe to be behind walls and do your research and not share so much because there's a little bit of, yeah, maybe fear of sharing or not. Yeah, they maybe don't want to talk about what they are doing so publicly. So I think it's important also to overcome this barrier there a little bit and also share a little bit of maybe of funding even because usually when you get a big grant, you get a little bit of money for outreach, and I mean, that would be perfect to give to an artist to work together. So yeah, I think that there's a lot of potential there still.
Yeah, and I feel like science communication is becoming a hot topic right now that, like you said, there's kind of this image of scientists being, hermits and working behind the scenes and, they do their 30 years of research and then, maybe not a whole lot comes out of it. But I think now there's this push for people to share that science a lot more readily and yeah, have these cross-disciplinary efforts in order to share what that is, whether, that's an op-ed in some article or having a gallery showcase. Because working with scientists, like especially in the deep sea, like what was surprising to me is how much visual data is collected. There's all of the biological data and oceanography data that's collected on cruises, but there's also pictures and videos, and there's terabytes of it. And so not only is it, great media to show, like I always think about Mbari and how they kind of make all these TikToks about little deep sea animals that they find on their cruises, but it's also really great content to inspire art. Um, like the artist that I'm working with for the artsy matchmaking is Meva Perez, and she's in Hong Kong. And she shared with me a bunch of videos and pictures from her cruise that I'm going to be making a poster out of. And it's really cool to see, these new habitats. And for me, it's the first time kind of working with an artist while not having actually been to that place. And I still feel like super connected to the project, and I'm really excited to talk about the research. So I do think, yeah, scientists should talk to artists a lot more because it's only going to be good for you to like show your work. It helps you get funding. It makes connections. It increases collaboration. Um, I think it's a great idea, and we should have more of it.
I also think you learn a lot more about your own study system, like just having this different perspective from a different person, seeing it more subjectively, maybe, which we sometimes are not really allowed to as scientists. Like we have to think in data and do stats and so on. So this is a way of breaking out of these like very strict boundaries.
Yeah. Yeah, we were talking a little bit before this about how, being both artists and scientists, it's really nice to kind of have that other side of your life. And, as scientists, we look with such a critical eye on these systems and our experiments and our data. And, when you're an artist, you're playing, like you're having fun and, those creative gears are turning. And I think it's really important because it also trains the creative side of your brain that you can still use in science also, coming up with new questions to answer, the best ways to show your data. I know I think a lot about data visualization and like the best way to show the results of my project without it being overwhelming. And I think being an artist and working with art has helped me be more aware of what how my science could be perceived also.
Yeah, so we definitely, so then that's, thank you for both of you, because, right, you have both science and art hats on, and you see the value in both. We just have to convince more people in the science part that, it's okay to share and collaborate, because we're all here because we all love what we do, right? Or after we learn what you guys do as scientists, I think artists always get very invested, and if we can shout it out to the top of the world, we will, however possible. And so I also want to thank you as an artist that, you guys are doing the hard work of both and trying to. So now we just need to convince our science friends to come out and play.
Yeah, and I would say the same for artists. Like, I wouldn't say it's just the onus of the scientists. Like, I think a lot of scientists, like, they're super busy, but if you keep bugging them or, reach out to them and say, hey, I love your science. This is the art I do. Would you want to collaborate? Like, that's also another avenue. Like, I know I was just in Antarctica in the fall, and we had an artist come down with us, and she actually like co-wrote a grant with my advisor in order to come down with us. And so I think there's also a lot of strength in being the artist and kind of approaching the science. Um, that's dependent on how public the science already is, but that, yeah, everybody can try to bridge that gap together because scientists also want, cool art to show their project. When they're giving that presentation at a conference, if you have this beautiful painting, people are going to pay more attention to it. Um, so yeah, I think if you're an artist too who wants to get involved in collaborating with science more, if you see a project that you're really interested in, I would reach out to that scientist and ask if there is potential to do some sort of collaboration. Maybe they already have funding set aside for outreach, like you said. And if not, you could kind of work together to work that into a new grant that they're doing potentially.
Well, I guess if we don't have any other questions, comments, or tips, thank you so much for everybody for being here, for joining us on Reef Chat.
Thank you for having us. I know I
Yeah.
haven't spoken up before now, but I'm really glad. This is the first time that I've ever seen one of these meetings. And
Oh,
it's
thank
really
you.
enjoyable. I hope to get in contact with at least some of you in the future. I'm
Thank you so
Eleanor,
much
by
for
the way.
joining. Nice to meet you, Eleanor.
Thank you.
Thank you so much for joining, everybody. If you want to learn more about the deep sea, there's always the Sustainable Ocean Alliance Deep Sea Academy. Thank you to Dexter and Lara for being with us here today. If you have any other questions at some point, don't be afraid to get in contact, and I'll make sure we'll get to them. And thank you for being here from your spot in the world. Good night, good
Thank
evening,
you.
good morning, wherever you're from.
Thank you.
Thank you
Bye,
all.
guys.